looks like the CP worked for everyone. At least it finished for a couple of people. So with that, I posted again the final command on that slide in the chat. So replacing your USR ID with your own Cyverse ID. So this one here. Okay. So hopefully everyone has a chance to run that. And again, this sudo chown minus capital R USR ID /scratch /botany 2020 NMG workshop. That /scratch/, as you can see on my screen, even though when posted in the chat, it might be in 2 lines, that /scratch /botany 2020, etc. is all one item. There are no spaces in that, okay? And so it's sudo space chown space minus capital R space your user ID. So in my case, it would be a Powell here. And then space again, and then /scratch /botany, etc. Okay, that should take no time at all. So you should keep your command line when you do that. And then we need to start up the Mazurka installation. So I can again post these into the chat box so you can copy them. And again, actually, if you widen the chat box, it maintains the lines as they should be as well. So we can do these. So first is cd/scratch. So here we're moving into the Scratch directory, which is the sort of main one where a lot of the work will be happening. That's where we have our disk space, as it were. So we'll do this git clone. So installing things from,
It's the error message.
Clone is not a git command. For people who were having trouble with git clone as well, just as I was, I somehow magically typed exit and relaunched my shell for Atmosphere, and now git clone is totally working. You can see it on my shared screen. So maybe that's worth a try for some of those folks. Yeah, so I didn't actually cover that, but to exit your shell, you can just type exit and close the window and then go back to here and re-click the open web shell and it'll relaunch.
It's like Ryan got Mazurka to install, which is good.
Yeah.
So
yeah. So if people completed and went to 100%, please carry on with these additional steps, and that that install.sh is sort of a key step. Yeah. The other thing I didn't mention that Cristiano brought up was that if you're SSHing from a terminal, as I indicated you could, using the IP address, you will be prompted for a password. That password should be the same as your Cyverse password, right? I didn't actually explicitly say that. So if you are doing or want to explore doing that, it should be the same Cyverse username and password, except you'll be doing it through the terminal. Okay, I'm just gonna power through these. So if people are still having issues with the git clone and Mazurka, maybe send me a message. We'll try and solve that. Yeah, Rebecca says the git clone command is now working, exiting and relogging in. Yeah, so hopefully that solves it for most people. and just, as I said, carry on with these commands on the slide for installing Zerka. I'll move on here, but you can hopefully follow that, download the PDF and follow it there. And the commands are also posted up in the chat. All right, so the other, so hopefully that Mazurka installation has worked for everyone. I guess for some folks that were following along with me and my troubles, it may not have finished yet. So please let that complete. I will talk through these Docker steps, and hopefully for those that are still waiting for it to the Mazurka step to complete, you can follow along with these later. And I can also post the individual commands into chat as well. So we're gonna also do a few installations using Docker. So our first step is this sudo service docker stop. And so I'm going to just paste it in, and we'll run that. And I gotta do this thing again. Okay, so it won't give you any feedback, but it should have been done. We'll do this command. You probably won't have to do this, or you won't have to do anything here, but we do this sudo emacs. I'll paste this here. Okay, and so hopefully you'll be taken to a screen that says something like this: brace bracket, close quotes, graph, or open quotes, then close quotes, graph, colon, quotes, slash scratch, slash docker quotes. So it should say this in that file. Okay. If it doesn't, please feel free to type it in. I guess, can I get a thumbs up, thumbs down of whether it does or does not say that for you? I can't see the participants. Where are they? Yeah, okay, thumbs up. So it does say that, hopefully for everyone. So to exit out of Emacs, hold down Control and X and C at the same time. So these three keys, hold them down and then release them. So hold them down at the same time and then release them. And you should get back your command line, okay? If that doesn't happen and you're trapped in Emacs forever, message us and we'll get you out. But so I held down Control XC at the same time and then released them. And now I have my command line back. So now we'll do this sudo rsync command. And I will paste that in here so you can copy it easily. And so I'll run this here. I'll run it with you. And this, I think, might take a little bit. I think the Docker config might not be relevant to us. It didn't seem necessary for me last time. But we'll get there once this one completes. Okay, so this has finished for me, so I'm going to carry on. I'll try this one, but it might not actually be relevant. Oops, let's not do that. And I'll post it in here. Got to switch to everyone. Yeah, command not found. This was what happened to me earlier. I don't think it's actually necessary in our case. So now I'm going to do sudo service docker start. I'll paste that in here as well, because I'm about to move on to the next slide. So even though I'm going to move on to the next slide, please just know that the preceding commands are also in the chat, and so you can copy them from there. Or if you have the PDF from the GitHub for this session, you can grab them from there. Okay, so for these commands, we're going to actually install the programs. So it's just that part, so not the second part after the hashtag. Is the command proper? So that. And it's just 3 of these Docker pull commands that we need to do. Okay, so I've got my portcullis from Docker, and auto.
I'm onto that where you add, where it has this graph scratch Docker thing.
Yep. There.
What do I do with that?
Right. So it says that, so it's fine.
Yeah.
You don't need to add anything. So just to exit out of that, hold down Control X and C for a moment and release them, and it will get you back to your command line. It will exit out of the Emacs program. So you should get your cursor back at the $sign command line.
I think it's asking me wait, am I still on?
Yeah.
I think it's asking me a question. Save file, blah blah blah, etc. Docker.
If it does ask you a question, I'd say no. It just means you probably typed something in. But right. We don't need to, as long as it said graph.
Yeah.
In there already, then.
Modified buffers exist. Exit anyway.
Yes, do it. It's a bold move, but we're going to do it anyway. All right. And so now you should be okay to proceed with the Docker steps, Rebecca. So, whoops, we don't want Mikado. And so I'm going to proceed and just install this last one, and then I'll hand it over to Jacob. For those of you who are maybe still working on the Docker stuff, I think we can keep that running and get you caught up, and just let me know if you're running into any issues. Just send me a direct message or anything like that. Okay, so I think with that, those were the last installs that we needed to make. Right. Okay.
All right.
And the commands will stay all in the chat there as well.
Get my stuff situated here. All right, before I go into things. If you go to the GitHub repository for the workshop, which I think is in the chat before, and I'll just put it in there now. If you look under GenomeAssembly, there is a BTI May 2021 PDF. This is going to be all of our steps for this next section. So download that. And I've got a few slides to go over as well. And those are going to be here, the GenomeAssembly slides. So I got just a few things, kind of an overview first, and then we'll get into the hands-on part. And if there's any questions at any point, feel free to put those in the chat or interrupt me. So for the genome assemblies and annotations, or for genome assemblies, actually, we're doing before launch. I was glad that Feli put something similar in his slides with the puzzle pieces. I couldn't find the same puzzle, but when we have, you know, a 1,000-piece puzzle versus a 50-piece puzzle, you know, how easy is that to assemble? And that's basically what we're doing with the genomes. I think Feli gave a very good overview of that. Here we have essentially Illumina data, all, you know, uniform, small pieces, many of them, versus some of these third generation PacBio nanopore, probably not as deep, definitely not uniform in size, but longer pieces, easier put together. And so here's just kind of a little more of a comparison with the reads themselves. You know, error rates are different, sample preparation is different, the amount of coverage they needed is different, but of course those are all things that Feli covered really well. But how it really impacts this section with the genome assembly, especially a de novo assembly, if we're working in a non-model group that has nothing close to compare, some of the major options that you have are either using Illumina only data for the assembly. So again, these are high quality reads, very few errors. However, your assemblies are often very fragmented but very accurate. They do have their uses, even though like you don't have chromosome scale assemblies, they can be very helpful for identification of SNPs, looking at for phylogenetic analyses or specific alleles. And so, you know, you don't have to have a great chromosome scale assembly to answer a lot of questions. Another option is a hybrid option where you have your Illumina data, and then you combine that with either nanopore or PacBio to get the assemblies. You can either use raw nanopore PacBio data, meaning what comes off the assembler, those are the sequencer. Those may be error prone or not. If you hit the Hi-Fi, they definitely would not be. Or you can error correct those before you move into different analyses. You can error correct those based on the Illumina data, which is more accurate. Or your third option, if you have enough data, you can use just the long reads themselves. This often will give you the best assembly, but you need to have the depth of the long reads to do so. And then you can, depending on, again, what method you approach, you can polish your assembly with the Illumina data to fix some of the errors that may exist there. You know, and so the recommendation, one recommendation for how much data you need is the 50x coverage of the short reads. You know, that could be a little bit overkill depending on what you need to do for that. And then the 50x long reads is an ideal situation. But that's for kind of like just some, you know, arbitrary numbers, but how much data do you need? Well, for the plants, specifically angiosperms, which what I study versus the species that Fei studies, our genome sizes can vary quite a bit across the angiosperm tree of life. The model species of Arabidopsis, 135 megabases, rice around 430 megabases, which is close to the mode that you see in the angiosperms. And then you start getting into bigger and bigger things. Ginger, 1.5 gigs, barley, 5.3 gigs. And then you get to see some of these monsters. And again, you know, if you're looking at 50x coverage for this, So for rice, that would be 25 gigabases of data. So it kind of depends on what your system is. So having a good idea of the ploidy and more importantly, the genome size will give you an idea of how much coverage that you need. Now, some of these genomes can be quite big, and so that can be a problem. So some assemblers were originally designed for the human genome or smaller, which is good for some systems, but for some of these crop plants, barley, rye, wheat, where you're talking 5, 10, 20 gigabases, it just doesn't work. The larger the genome, oftentimes that extra material is repetitive elements that are harder to sequence, but your number of genes are roughly the same in most cases. And so those repetitive elements are definitely almost impossible to assemble with the short reads. The long read data really makes that possible. But then you have computational resources can be intense, especially for large genomes, repetitive genomes with the amount of data you need. And that computational resources is either in terms of memory to run it for RAM, storage requirements to store the associated files, and wall time, the amount of time it takes to run an assembly. And so some of the different options that I've mentioned. So one that we're redoing today at Mazurka, this is a great option for both short read data, and if you have hybrid data, meaning that you have your short reads and your long reads together, but maybe not a whole lot of long reads. And so this is a very fast, accurate option. Newer versions are always coming out compared to other programs previously compared against. It can produce longer in 50s. So kind of what's your middle point of the size of the contigs, and more of the BOOSCO genes, or those benchmarking single copy genes that should be there in most species. It's a way for us to kind of get an idea of how complete our assembly is. And so Mazurka combines the Bruin graph and overlap layout consensus. So I mean, this is very confusing. What does it mean? So kind of roughly, what Mazurka is going to do is you're going to give it either your Illumina and or your long reads. With the Illumina data, it's going to try to kind of scaffold these together and create these super reads. And then it's going to try to overlap the super reads from your Illumina and your long reads from PacBio, merge these into mega reads, and then try to link these if you have additional data to do so. Incorporating that long read data helps a lot. And so here's just a comparison of the PacBio coverage going from 0x coverage of long reads to 60x coverage, and the N50 config size that you get. So again, these are just building the contigs. It is not incorporating how many contigs you have, but when you go from 10 to 20x coverage of PacBio, your N50s are almost 5 times as big. So a little bit of long read data can do a lot to help with your assembly. Once you get to the point of 50, 60x coverage, at least in my experience, Mazurka is not the best option then. Some of those long read only assemblers will work. But if you have a bigger genome species that you're working on, it's harder to get to that. So Mazurka can be a great option for a lot of things. But how much, what are some of those computational resources that you may need? And so here's an example from 3 different species. One, Utricularia gibba, which is what we're using today. It's got a small genome, only 78 megabases. For this, we have 120x Illumina, 100x PacBio. If you include all that data, to assemble the genome, it takes about 3 hours with 16 threads. So that's not too much. Storage is minimal. If you go from where we are here to a little bit past the mode, say for a 1 gig genome, this is again some of the stuff that Febe was mentioning. If you have 80x Illumina data and say 20x nanopore data, again, with the genome size about 1 gig, it may take you 3 days of assembly time with 28 threads, and your storage is now 200 gigs. Now, if you go to something bigger, say around 5.5 gigs, even when we don't have much in the way of nanopore data, this is an older run I did with 40x Illumina data and 4x nanopore data, this itself took 22 days, almost 4 terabytes of storage and 28 threads. So, so as you get bigger and bigger genomes, the amount of sequencing data you need and the computational resources behind that to assemble that goes up. So just something to kind of keep in mind as you're thinking about your own projects, what you may or may not need computationally wise as well. We talked about Mazurka using Illumina and hybrid only, or the hybrid option. If you have enough long read data, PacBio, Nanopore, these long read assemblers can work really well. And there's many different options out there, different flavors, different ways to do it. In a lot of cases, the size of the genome may make or break the different options of what you want to use, how well they work for your system, the heterozygosity and whatnot. 2 options that we'll talk about today and that are in the tutorial are Fly, which uses repeat graphs. This one I found, at least for around the 1 gig genomes, to do really well at assembling in terms of those Busco genes. Another option is this WTD-BG2. So it uses a fuzzy Bruin assembly graph. It is much faster. However, I found that the outputs, the results are not quite as well or as good, but I wanted to include that as well so that you can see how it is done and more importantly, how fast it can be. And then you can compare that to the other options. So, there may not be just one assembler that works for every single thing, for every single project. So trying multiple ones can do well for you, at least to see how they compare. I mentioned a little bit earlier, and Fei did as well, about the error rate for Nanopore and some of the PacBio. Can it be a problem? It definitely can be, but both the 3rd generation technologies have made vast improvements on their error rate. Neither of them are quite to Illumina standards, so I think the Hi-Fi is getting close. So how do we fix this? Is this having this error-prone data bad? Not necessarily, if you know how to address that. And so there are 2 ways to do this. One is to error correct prior to assembly, which can work very well. The other is to assemble with this raw data and then polish with your Illumina data afterwards to replace any SNPs, indels, changes that may have occurred. The Illumina data for these methods are useful, very useful, but there are other options for error correction without Illumina data if you don't have access to that. So for the error correcting, there are 2 major ways to error correct. One is a non-hybrid approach, which is basically just correcting based on depth of the long reads. So let's say we have 3 reads. We see that each of them has an error someplace, but the other 2 do not. So based on the consensus rule, you can replace those errors as we see here. So now you have 3 clean strands. For this, the more depth you have, the more likely it's probably going to work. There's also some K-mer filtering. If you have a certain repeat that only shows up once versus thousands of times, such as in WTD-BG2, it will just remove that. That's problematic if that's actually a biologically relevant K-mer. But again, the depth of the sequencing makes a difference there. The other option is hybrid, which uses that Illumina and long read data. And so there's several different options there. One is alignment based. So you align your short reads to your long reads. If there's places that may be fixed, it corrects those. Another one is a kind of an assembly based, using an index of repeats as well. There's lots of options to do that, including FMLRC, which is one of the ones that we're going to be using today. For the error correction, especially with a hybrid approach, it's been shown in several comparisons to work better. The Illumina data is much more accurate, but not all of these error correction programs are equal. There are several factors to consider when comparing those, such as the sensitivity, output rate, alignment rate, accuracy is a big one. The efficiency in terms of memory and time. Both of these things can be critical for trying to figure out what is the best way to do with different study systems. And here's just a comparison between 4 different methods that show you that sensitivity and efficiency may change. Accuracy for some of these is pretty good, but not always. And so, you know, again, there's not necessarily one method that works for everything, and there's always new methods coming out. And so what we may use today, you may not use 6 months from now because there might be a better option out there. So it's always good to keep an eye out what some of the options are. So this is for error correction, usually before you do the assembly. There's also plenty of options out there for polishing the assembly after it's done. And so the one option that we're going to be using today is called, it's a Polka as part of the Mazurka package. So you'll assemble your long reads using Fly or something else. And then what you'll do is you'll take your short reads, map those back to your assembly, sort the position. So you're using BWA-MEM, SAMTools, and then you'll essentially call SNPs with freebase and indels, and then replace the high quality variants with the low quality score on the assembly to correct some of these issues. Again, there's many options out here, time, efficiency, all things to keep in mind. But Polka has done fairly well for me in the past, and it runs very fast. So we're going to give that a shot today. Just a couple of other quick things before we get into the hands-on. I've mentioned this N50 value. You'll see this a lot in the assemblies that get output today. So basically the N50 is just where half of your data is at least a certain size, and this reads or contigs. So in this case, the N50 is 14,000 base pairs. You can also have other ways to talk about that, but usually the N50 and the number of reads or the number of contigs are 2 of the things that you'll often see describing how complete a genome is or how good the assembly is, with the other being BUSCO. So these benchmarking universal single copy orthologs. It's a way to address or to assess how many genes that we think should be there in a given set is in your assembly. Now, these are not always 100% accurate. Some of the BUSCOs are actually duplicated on some species, but it is one of the best ways we have to assess quality. And there are specific libraries for certain groups that you may be working on. So one is the Embryophyta. So mostly it covers most of the things for the angiosperms. But if you're working on groups such as Brassicales, Solanales, Poales, there's going to be a subset of genes that will be found in all of these orders. And so if you're working on those are often better things to use. And so with this tutorial that we're going to walk through before we go to lunch, we're going to be using the Utricularia gibba. It's the hump bladderwort. So it's a carnivorous aquatic species, small genome size, which is great for running things quickly. We've also subsetted the data to make it run even faster. And all of the data that was originally built, or that this workshop was built on, is all publicly available data in the SRA and other repositories, as well as some RNA-seq data that we might be using later this afternoon for looking at some downstream analyses. So, you know, not something that we all work on normally, but a very useful system right now for this workshop. As we go through the tutorial, you don't need to fill this in, but if you want to, you can, we'll pay attention to some key things, some of the different assemblies, such as the number of contigs, the N50, the size, and how many variants are polished. We won't run through all the BUSCO things. There's the code at the end to do that, but those often take a little bit longer. But if you want to run through, you can see for these different assemblies, how many BUSCO genes are present. Any questions before we get into the hands-on part? No? Okay. So let me see if I can. If you have the PDF opened up all of the codes that we're going to be doing are on there. So you just copy and paste those. I think most of you have already downloaded this older version of Mazurka, installed that. This is necessary for the Mazurka hybrid approach, where we're using both the Illumina and PacBio data. If you don't have it now, it's not it's not too bad. We'll use the pre-built version of Mazurka, which works great for the Illumina data. So there's only one step that you will not have. And then the data gathering, you did this with Adrian earlier. So this is where all of the data is going to be. So if you're trying to work on this on your own, or need to remount the virtual machine, this is one way that this could be done to make sure you have all the data. Now we do have quite a bit of data, and so we're not going to use all of it for this, because otherwise these analysis may take several hours versus several minutes. And so the first thing we're going to do is do this data subset. And so if you copy in the commands into your terminal. So for me, I am just now moving to the folder in Scratch that is the raw data with all the genome data, and I'm going to subset what is there. So these next two commands are going to take the Illumina data that's there, unzip them, and then take only the first 400,000 lines of data, so the 100,000 reads, and create a new smaller FASTQ file that we're going to use for all the different steps later on, just so it runs faster. If at some point you want to go back and redo these analysis with the unsubsetted data, you definitely can. It'll just take a little bit longer to do. So if you copy paste the zcat, this is one of the sequencing files, and it's going to pipe that in, read the first 400,000 lines, and it's going to create this polish test R1 FASTQ. We'll do the same thing for the second one, and these two should run very fast for you. Second part is to unzip the unzip this PacBio data, and hopefully why are you not doing it? I'm not sure why it's not working for me. Hopefully it's working for others. Let me make sure that it's there. Okay, mine was already unzipped. That's why I wouldn't do it. I did that yesterday. So you should unzip it. It may take just a few moments to unzip. And then once you see in the raw data folder, you should see this. You give a PacBio to use. Now it should be .fastq. And then once you have that, we're going to subset that again with head to take the first 200,000 lines. Are there any problems so far with getting the data subsets ready to go? I'm going to take the silence as that we're good. Taking long. Okay, yeah, it might take the unzip part of, especially the PacBio, is going to take the longest time, but it shouldn't take more than a couple minutes. So I'll pause just for a second to make sure that everybody else has completed that. So if you do the LS again in your folder, what you should see now is once you've got through these different steps, you should see polish test R1, polish test R2, the original Illumina data files 1 and 2, the original PacBio file that you give a PacBio to use, and now the UGIBA PacBio subset. So you should have all 6 files in your raw data UGIBA genome folder. Has anybody gotten to the all 6 files created, or all 6 files listed still in the unzipping part? Okay, so that one person's, a couple people are done, a couple people are still unzipping. If you have got the unzipping and all this done, but I'm not going to move on to the next step completely, but we also want to make sure that we have one of the programs that has some issues being installed previously, Jellyfish, and to make sure that it's installed. So if you copy this sudo apt update command, you may need to type in your password. And I got to make sure I actually select the entire thing. So we're going to make sure that we have things. We're going to do that, and then sudo apt install Jellyfish. Just want to make sure that we have Jellyfish in our path so that it can be found. And so mine was already installed and updated, so we're good. If you haven't done that yet, you'll want to make sure that you have that installed as well. So it looks like a couple people now are getting the files unzipped and subset. That's good. Everybody's still progressing nicely on that. Looking good. Okay. So now that we're ready to hopefully start doing some of the assemblies, we have our test data sets. The first one we're going to do is use only the Illumina data we have for Mazurka. And we're going to see what that assembly looks like. And so we're going to follow it along. First, we'll move back to scratch. And then we will create a Illumina only, a Mazurka Illumina directory. And so I already had one set, so I'm going to delete that. And so if I look here in mine, now I have Mazurka Illumina. I'm going to navigate to that. And if you do ls in this, it should be completely empty, which we just created that folder, so that's not surprising. We have the configuration file that we need for Illumina in the botany data set. And so we're going to copy this over. Nope. And so that's what the scp command is. Or you could just do CP, but it's going to be there now. And so that is in our file. So now we want to just look at that. And we're going to use cat just to look at it for the moment. And so if you do that, it's just going to show you what this file is. And so this is kind of the standard configuration file that Mazurka packages with the GitHub download. It tells you what needs to be done. But essentially what you can do, you're going to specify where the data is. That's what this top part is here. And so for us, we have paired in Illumina data, which is one of the things that you must have. So you tell it it's paired in, what the fragment size is, and if and kind of what the overlap may be. This is kind of default from what was given. with the example data set. But the key thing is to tell it what files you want. And so here we are telling it the polish test R1 FASTQ and the polish test R2 FASTQ. If you have aluminum mate pair data, you can give it pack bio data. If you have it, you tell it where the path is. We will be doing this, but not quite yet. That's a little bit later on. If you have nanopore data, you tell it what you have and any other options that you want to give it data-wise. For other reads, if you have Sanger, 454, whatever, you can specify, you can give it here. The bottom part of this file is the parameters. Most of what we're going to be dealing with is going to be the default parameters that are specified. You can change these with your different runs if you want to, but we're going to leave those the same. The one thing that we're going to change, though, or that we can change, and that you would want to do this, is the number of threads. So for our virtual machines, we're only using 6 threads because we only have 8 threads total. So it's going to run through about as fast as we can allow it on our machines and still have enough to move stuff around if we need to. But everything else is going to be kind of default. If you look at the user manual of Mazurka, you can kind of learn more what some of these things are, if you need to change it, if you should change it. But it's going to be depending on what kind of data you have for your particular project. And so now that we know our configuration file is set the way we want, so run Mazurka, it's 2 lines of code. The first is to specify, to tell Mazurka where our configuration file is. So we're going to launch the Mazurka package or the Mazurka binary that's in the 4.03 package, and we're going to tell it what the configuration file is. And it's going to go through, and it should be verifying paths, is jellyfish installed okay? Are these other things okay? If there's something wrong with the setup or the installation, it will tell you here that there's a problem. So you can go back and fix that. But if it tells you everything's okay, and it says execute assemble.sh to run assembly, so we're going to execute that file here, and we're going to progress through our Mazurka run. Are folks seeing this? Have you got to the point where Mazurka is running? Are there errors that you are getting when doing this? I see a couple people.
Yeah, I think mine has like a different version on the slides.
What do you have? So you should have. What does it say there?
My slides say 3.4.1.
So that is so did you download the BTI May 2021 PDF, or do you have one of the older ones?
Huh. Let me check. No, it's the 2021.
So,
oh no, it's the other one. Yeah. Okay, my bad.
So no, it's okay. So the opt will be the same. The Mazurka part here will change based on which version's installed. So if you have a different version, the slides may say the 3.41, but if you just put in the 4.3, everything else should be fine.
Okay, I'll try that. Thanks.
Okay. All right. Was there a question? So there's a problem with the no directory. What is going on with that one?
It just says that no, when I run that code, it says no such file or directory, but I created all the other.
Is it the opt Mazurka or the .assemble or ./assemble? When you do the opt Mazurka, what is that the one that's telling you that there's no directory there?
Yeah.
So it's having trouble finding the Mazurka binary. The other option for this is if you downloaded and installed the new Mazurka, or not the new Mazurka, the old Mazurka, which was Mazurka 3.42. If you do this part, so instead of opt Mazurka 4.03, if you do scratch Mazurka 3.42, bin Mazurka, and then the SR config, if the rest of that is the same, then that should run. Okay, so scratch Mazurka opt. No. Oh, okay, so yeah,
there is an error when I run that. There's at the beginning of there, it says that error usage, scratch Mazurka.
So I think the problem is a version control. It's a version problem. So if you gave it the opt Mazurka 3.41, that's the one that's I think the old. That's not the version that's currently installed. If you do the opt Mazurka 4.03 that Maria posted into the chat, that should get it to run based on the current settings. If you get Mazurka to launch, initiate, and to run, based on, because we're not giving it very much data, it runs pretty fast. And so in my case here, here it spits out the some of the summary statistics where our N50 is 18,000 bases. We have assembled a total of 158 megabases of assembly. Our average contig is 1,700 bases. And then our count, so we have now 92 contigs. It worked. Okay, fantastic. And so again,
I have a question, a quick question.
Yes.
My numbers are very close to yours, but not exactly the same. Is there a reason for that?
It's actually a very good question. I know it should be very similar. I don't actually know if this is probably an issue with so I don't know how, if you give the exact same data to Mazurka 10 times in a row, I don't know if the assemblies are always going to be exactly the same each time.
I don't think I would expect them to be. I think there's some element of randomness in the assembly algorithm.
Yeah, so I think as long as you're close, I mean, that's a good sign. But I've not sit down and tested it to see how much variation we should expect.
I am close, but just not exactly the same.
Yeah, I think as long as you're close, I think that's good. But that's something that I should think about and try at some point as well, actually, because that could be an interesting comparison. So checking into the chat, Rebecca, you said that you're having trouble still.
Yeah, it's
okay.
I think it's got a problem with a config file.
Yeah, so I think if you make sure that you do if you make sure you do this copy the config file from one of the other directories, because make sure that's in there, because that's the problem. I think no such file. Yeah, so it looks like Mazurka is finding Mazurka and trying to run, but it can't find the config file. So that's a little bit different problem, I think, than and hope that's easy to solve. I hope. And so when we get to it, the hybrid assembly is going to be a very similar process to this, but we're going to tell Mazurka that we're going to use some of the long read data. But before we get to that point, I want to compare what we have here to if we were to just assemble something with the long read data only. And so for this one, we're going to do the fast option with the WTDBG2. And so here I'm just going to go back to scratch and create a working directory, and then move into WTDBG2. Now it should be empty. And so what we're going to do, instead of copying files over this case, because we don't really need to tell, we don't need a configuration file here. We just need to tell what the reads are. And for WTDBG2, it's 2 lines of code. The first one is going to be here, where basically we're going to call the program. We're going to tell it that roughly kind of what we expect to see. Genome size here, we're going to estimate a little bit bigger, but it's going to be fine. And we're going to tell it where our PacBio data is in the FASTQ form. And then it's going to use 6 threads. Oh. And I think there was a problem here with line options. So let's see if I can do that. So if I put in that in the chat, is it going to actually, it does seem to be maintaining low line breaks. But of course, it doesn't want to do it here. Let's see if I can do. Yeah, so I had to just edit that to make sure there was no line breaks. Here's the new one that should work fine. And so now it's going to go through the process assembly. It's going to look for those really low k-mers, remove those if they think it doesn't exist, and try to stitch together a assembly here. And again, it's going to go very fast. So what we have here now is part of the assembly. We need to do the second line of code as well. So it's going to finish this off. But up to this point, what we had though was 1,300 contigs with a N50 of 24,000 bases with a max of 120,000. But this is only partially through the assembly process. Here it's going to start matching those contigs up to make sure it can find and to get rid of any redundant ones and see where things are. But already we are having a bit better assembly than what we had beforehand with Mazurka, with just the Illumina data. Now, is this part working for folks with that correction with the PDF? Sorry about that line break. That's always a pain. And so again, this option, it uses that fuzzy De Bruin graph. It's going to be relatively fast. How accurate is a different story. Again, it's not one I've used this before for other projects. It's one I don't typically use now. I don't necessarily trust. I don't think it's quite as good as other options. However, it is super fast. And if you need just a quick test run to say, how's my data looking? What can I expect? This is not a bad option for that. But there are a little bit better options out there. The next ones being fly, which we're not going to do today because it takes longer to run. It may take a couple hours to run the same data set, but this is just showing you what the commands in that black box, what you would run if you want to try fly out at some point later on. All of this shall work. You're going to make a new directory and you're going to basically just call fly, tell it you have the raw pack bio, where those files are, and the output directory, and the estimated size of the genome. Again, this is where the estimated size comes into play. Again, we're not going to run this now because it does take a little bit too long for now, but it is a good comparison and a bit more accurate. And so I think we had, what, 1,300 contigs total. So we're going through now 700. We got a little bit left to work through, but it is going relatively fast. And it'll be interesting to see what the final output looks like in comparison to what we were getting for Mazurka earlier. Okay, I see, Rebecca, your comment, the ls command shows, let the scratch. All right, so if you do the ls in the Mazurka, so this is from Mazurka still, right? So if you're doing that, that means the file is there, but why? Oh, well, I saw that at least in your at least in the opt command that you had, there was this extra 7 after the text. I don't know if that was just typing into Zoom a problem, or if that was what was actually in the command. If it was in the command that you gave the terminal, that could definitely be a problem because it can't find the Illumina only .text 7. It should be just .text. I don't know if that's going to solve the problem or not, though.
Okay, I'll have a look for that 7. I don't remember typing.
I mean, I'm just saying what I just saw that in the chat. I don't know if that's.
But then that's after this one. Like that opt command is after this.
It is, but I mean, if you have the STP scratch, but if the config file is there, it should. Then the Mazurka opt should be working. I don't quite know where its issue is. Maybe, yeah. I mean, we're gonna go back to Mazurka in just a moment or two.
I put a W when there was supposed to be an N.
Typos, they get us every time. I mean, I don't know how many times I've had commands that fail because I typed something in wrong. So yeah, do not worry about that at all. We've been down there a lot. All right, so I just want to look at my WTDBG2 output. It looks like have assembled 1,300 contigs with a total basis of, what is that? 24 million. And so we know we're not assembling the entire genome. About 78 million bases, I think, is what we should be getting. Again, we're doing a subset of the data, so not too surprising. And so that's just one assembly to compare. And so again, we're going to skip over the fly directions now. I want to get a little bit into the error correcting and polishing aspect. So this is, again, the error correction is before assembly, polishing is after. And so for the error correcting, we're going to use a program called FMLRC. So that's FM index long read correction. It takes an MSBWT. I know there's lots of acronyms here. An index for that for the multi-string Burroughs-Wheeler transform. That part can take a long time depending on the size of your data. So we already have that made for you. But if you had your own data, what you would wind up doing would make this this transform data set with this command, or substituting in your appropriate Illumina data. Again, we're not going to do that now because it's already been done for us. But just so you know, if you're doing this on your own, that's what would happen. What we do need to do now is to actually, now that we have our index made, we want to use our Illumina high accuracy data to error correct the potentially problematic PacBio long reads. So if you create and move to, we don't need to create it, you just need to move to this folder in the genome corrections or GenomeAssembly error corrections file folder. You will see this transform file that we're going to use to error correct our data or long read data. And so the command to give this is here, where you're going to call FMLRC and you're going to tell it, line breaks again. Let me fix that. All right, so this should be in the chat. Let's hope this works. And so at this point, where's my problem? Okay, I don't know what happened there. Story of all bioinformatics, it seems like. So for this correction, let me make sure I have this correct here. So what we're going to do is we're going to call the program FMLRC2. We're going to tell it where our transform array is, and we want to tell it what the input files to correct are. In this case, it's this subset of PacBio that we used earlier. And then the last part here is it's going to create a new file, and it's going to error correct the FASTQ files and create a FASTA file. Now, the reason it goes from FASTQ to FASTA is because once we error correct, we'd lose the quality score associated with the FASTQ data. So all we have is the FASTA file. And then here we're telling it to do 6 threads. And so it's going to tell us here basically what that was. It tells us where that transform set is. We have our input reads and what the output should be. It's going to go through and give you some more information. And so it's loading it's loading the compressed values from that array. So this is going based on what the Illumina data says. And it's trying to look at the FASTQ file against those arrays to see where things do not match, and then fix the low quality ones to the higher quality score. And now it is starting to process the reads. It's going to do those in batches of 10,000 reads, or it's going to report back in 10,000. And so here it finished processing about 50,000 reads in total. Again, if you had your full pack file, your full nanopore, you'd have a lot more than that. But now we have, if you do LS here, now we have this error corrected FASTA file ready to be moved into either long read assemblers or hybrid assemblers with that error corrected data that we have a pretty high confidence in of how correct it's going to be. Everybody okay so far with that? I got some thumbs up, some nods. Good deal.
I'm getting something when I try to do the optrust-fmlrc command. I am being told I need to provide more files. So I'm having trouble here. Are these 3 lines right here underneath the CD scratch, the starts opt rush,
Yeah, so
and that, is that one long command?
That should be one long command. There's some line breaks in there. So if you look at the chat, I try to get rid of those line breaks and copy that in. There should be one long file, should be one long string, where you have basically specify the MSBWT, the input, and the output.
Okay.
And so again, this was the correcting for the errors ahead of assembly, but you can also correct for the errors after assembly with polishing. And for this, we're going to use that Polka, which is part of the Mazurka package, which just stands for polishing by calling alternatives, or calling variants essentially, using the short read data. For this, there's a couple of things that we want to make sure that BWA is in our path. And so if you do, again, similar to what we did earlier for jellyfish, the sudo apt update and sudo apt install BWA, you should be able to make sure that it's downloaded, installed, and ready to go. And once that is ready to go, you're going to make a new directory called Polka Polishing and CD into that. Now, before we can polish, we want to make sure we copy our file. We're going to copy a pre-assembled version of the UGIBA genome that we assembled with Fly earlier. Make sure that's in the path that we are, so that we can align our reads to there and polish. Where did my, I'm missing something there. For the CP secure copy protocol, yeah, I was missing a period. So here it's just going to tell us we're going to copy the completed assemblies UGIBA fly assembly.fasta file, and the period just means we're going to copy it here and put it wherever we are, which we should be in the Polka polishing. And so I'm just going to take a quick look. I have run this before, but I do have this UGIBA assembly FASTA file. Once that is set, I'm just going to correct these line breaks again. I'll put this in the chat. So this is the command for the polishing. what we're basically doing here, and this is going to go over, for me, it went very fast, but we're telling Mazurka, particularly Polka, that our assembly file is, in this case, the uGiveAFly assembly. We're telling it where our Illumina data is based on the -r to -reads, and that's going to be our forward and our reverse polishing files that we had. Again, how many threads, and then how much memory to give for Freebase to run this. This should be pretty fast in this case. For larger genomes and large amounts of data, it can take longer because it's going to map all of your Illumina data onto your assembly. But if it works, it's going to tell you here at the end the substitution errors that it found, the insertion deletion errors that it found, how many of those and how many of those it fixed, and then what the assembly size is. So in our case here, our assembly size is about 85 megabases. And this is different from what we saw earlier with Polka, or not Polka, with WTDBG2. But now we have a polished assembly that you can see in our folder, which is this. So Polka just to the end of the name appends a .polka corrected .fasta file. So this is now our error corrected or polished assembly that we had from a previous run that we can then use for downstream analyses. Did that work for some folks? The polishing should be very fast. Still running, okay. As long as it is running, that's a good sign. That means things are working and the errors and the line breaks on the code are gone. And so I'm not going to move on to the next step quite yet, but with the error correction we did earlier, why do we want to do that? I mean, so part of this is, and for the hybrid approach, when we want to assemble the genome, we can take those error corrected reads now and input those into Mazurka, and that's going to help really tie together some of these contigs and make these larger kind of scaffolds. And we have high confidence that those are going to be accurate, which will make that matching up a lot easier to get a better assembly for our species of interest.
I have a quick question.
Yes.
Are we supposed to have all those files in this Polka polishing, the ones that you have there, right?
Once it's done, once it's finished running, you should see all those. To start with, all you really have would be this. All you have would be the uGiveAFly assembly.fasta, because that's what we are copying over here. And I'll put that in the chat because I missed here. The period is missing to tell it where, or actually it's not. It's the scratch of Polka is where it should be, and so it should be. Yeah, if you put this in, it's going to copy the uGiveAFly assembly FASTA to your scratch Polka assembly or Polka polishing. And then once you have that, and that should be the only file you have first, then once you run this line of code, which is in the chat, then it should create all these others. It's first going to align with BWA, sort those, then call variants, and then you should see all of these extra files at the end. But the most important one is this Polka corrected.fasta.
In my case, I got an error saying that BWA is not found on the path.
Yes, and so in that case, make sure you do this sudo apt update and sudo apt install BWA.
Okay.
If you do that first, then when you run the polishing, it will be in the path ready for you to go. And so I know we have about 5 more minutes or so before lunch. I don't want to keep everybody too long. I know everybody wants that free lunch, whatever it is downstairs or in your kitchen, wherever you may be. The last piece to do for the assembly is that hybrid approach. And so if you had problems with Mazurka earlier, hopefully we can fix all those problems. However, I will say that this is where that new installation of Mazurka, the new installation of the old version of the program is going to come into play, because we're going to be needing that. So if you if you have your terminal back open or back where you have commands to place in there, you're going to go back to scratch, create a Mazurka hybrid folder. I'm also going to delete what I had in there beforehand. We're going to delete that. Create the folder, move into that new hybrid folder. And then we need to copy again the configuration file, but it's a different configuration file than what we use for Illumina. So this is the hybrid version of that. And when I copy that over and I look at it, it's going to be the exact same we had earlier, except for the change is that we now specify where our PacBio data is. Everything else we're going to be doing is exactly the same. And so when you go to run that, instead of the opt Mazurka 4.03, we're going to use the new the new installation that you did in Scratch. And this should then tell you that, the mega reads assembler is okay, everything else is okay. And then you can go ahead and run this. And so it's going to go through a lot of the same steps. It's going to try to error correct your Illumina data. It's going to try to make these super reads. Then it's going to use this long read data to make these mega reads. If you had Mazurka running fine earlier, this should be no problem for you, because I hope that's the case. The older version of Mazurka is a little bit slower than the newer ones, but this part works better with this version for some reason. And so if you were to this point, and I don't mean to, I'm not trying to rush too much, but I just want to make sure that everybody is kind of in a position where you kind of see what is happening before we head to lunch. It's made those. So here it is using the 25x longest reads that we have for PacBio. So it's not using all our PacBio reads, just the longest ones, making these mega reads, joining the closing gaps. And it's working on assembling those now. And again, because we have a little bit more data than last time, it takes a little bit longer. And it's finished now. And so now what we have, if you remember earlier, we had what, 14,000 base pairs as our N50. Now we have 140,000. We assembled, what was it? Not great. What did we assemble here? Well, we have four contigs now, with the average of 44,000. And so we are assembling some things. Obviously, again, more data. If we use the full data set, it'd be better, but it takes a little bit longer to run. But now we have our final assembly FASTA file that we can use. Okay. Yes. Leah, for the chat, try using, instead of Mazurka 3.41, try using the 3.432. Let me see if I can get this in the chat. So that if you installed Mazurka earlier, if you did that installation that we did earlier, that code I just put in the chat will work for you. If you did not install Mazurka, the pre-installed version will kick back an error at this point, trying to use the long reads. Again, I don't know for sure why that new version is having so much trouble. But this should, and I didn't send it to actually everybody. Okay. So yeah, if you look at this, what I just put in chat, that command will work. Okay, Armando, I think if, yeah, the sudo apt install BWA should have fixed that.
I tried that, but it's still the same. Maybe I did a mistake.
Susie, do you have a suggestion for that? Because it should, with the sudo apt install, it should install the program, and it should be in the path in your path automatically.
So what's the error message?
It says a BWA not found on the path.
Even if you put the whole path to it?
Well, that's the problem. It doesn't, with the Polka, it assumes BWA is in your path already. And so that's why I put in the sudo apt update, sudo apt install BWA.
Okay. Yeah, that should work. The other thing I guess you could try is taking the BWA executable out of /opt and symlinking it to user local bin. But if you're installing it through apt, it should. So if you just type BWA, what happens? Just nothing. It says not found.
It says not found.
And you did the sudo install?
Yeah.
What if you type /opt the path to BWA? So it's opt BWA probably.
What did you write it?
So what happens here? I'm going to put it in the chat. So if you just type that.
Oh, it does appear. It says BWA.
All right, so try, hold on, I'm going to give you another command to try. You can try going to user local bin, and then once you're there, you can try symlinking it there. So that's the directory where the system typically looks for executables by default. So that may work.
Okay.
So after you do that, try typing BWA again, nothing else, just BWA, and see if it executes.
Well, it does something.
Oops.
But I have to change the directory, or it's from the local bin.
So just go somewhere else and just type BWA. Like go to your home dir or whatever.
Yeah, now it does call it.
Okay, so maybe try the whatever you were trying to do again and see now if it recognizes BWA being installed in the location that it wants it to be.
Yep.
as we kind of troubleshoot things, we're mostly done. I just want to point out 2 quick things. For the quality control, there's some things that you can do to look at your assembly. One is to look at nanoplots, which will plot a distribution of your contigs, tell you your N50, your assembled bases, and so forth. So if you use, this is for Docker, so you do need to make sure that if you want to use this, that you do the Docker pull nanoplot. And then if you run it here, this will look for, after you do the copy of the assemblies into the new folder called as complete assemblies, you can go through and test each different assembly that you have just by swapping out the FASTA name to whatever you want to, and they'll make some plots, so you can look at those and plot those. And then the second part is BOOSCO, so to generate those BOOSCO genes, to saying what works, what's it looking like. If you do all this command, it will generate those for you. You could plot those. Normally you could. There's a problem with R in this installation where you can't make those red, blue, yellow figures that we had earlier in the slides. But running this BOOSCO will tell you how many single copy genes you have, how many are fragmented, how many are missing completely. And so these are things that you can test out a little bit later on to get an idea of how your assemblies worked for the different programs. I know it's about lunchtime. Are there any other questions that we might be able to address, help troubleshoot? If there's parts that you didn't get a chance to do, but if you want to, you can go back maybe later and rerun those. I think most of the stuff should be there. I know we hit a lot of issues throughout things, but I think we have a lot of those sorted. So hopefully it works better now.